Good afternoon class. Today we are going to study a topic of immunology that is covering your immunology syllabus SEM IV of zoology, unit 10, that deals with unit that is the types of vaccines and active and passive immunity. So let's start with the introduction. So vaccines. So as we all know, recently we have gone through the COVID situation. There we have known the importance of vaccine. So they are the biological agents which contain a preparation of pathogens. Either they are killed or they are in the weakened form. Okay. The main motto of vaccine is to induce immunity and which have an long lasting memory. They are made from weakened or killed microbes. Another thing is that vaccines are meant for early recognition and destruction of microbes by host immune system, and it should also be cost effective. It can be of two types. One is the prophylactic type, that is in order to prevent a disease we can monitor vaccine, or it can be also therapeutic. Okay. So, and we will see that it has contributed to a remarkable improvement in the healthcare sector. So, let's move on. Here we will see a historical background from where does the vaccine came from. So, the first thought of vaccine actually came during the 1780s when Edward Jenner came with the development of smallpox vaccine. So that went on, and then Louis Pasteur also developed vaccines for rabies, anthrax and many more such diseases. And then polio vaccine was developed in 1950s, and if we see the scenario now, like the smallpox has been completely eradicated. Okay. So this is a small introduction or historical background of vaccines. So in any kind of diseases, our body cope up with the disease in two ways. One is the active immunity, another is the passive immunity. In case of passive immunity, the immune system doesn't grows antibodies by itself, but maternal antibodies are given, that is the antibodies are given from outside. Either it can be given from maternal source, that is through colostrum, that is the first milk that is given to the baby after the baby is born, or during pregnancy when the mother is antibodies are given through the placental, that is maternal antibodies, where ready-made antibodies are given. And it can also be artificial source. Artificial source means anti-venom, like in case of snake bite. In that case antibodies are ready-made. Okay, so that is passive immunity. And active immunity is one where the body is growing its own immune system, or the body is growing the antibodies by itself. For example, if we come in contact with any natural infection, we are going through any cough and cold or any cuts or infections, in that case the body is healing itself. So that is natural infections where the body is growing antibodies by itself. Another, it can be from artificial source, like any kind of toxoids, as we know tetanus toxoids. So in that case, or tetanus toxoid, also a very good example of active immunity is vaccine. Okay, where we are giving dead microorganisms or weakened microorganisms, and then the body is growing its own antibodies against that microorganism. Okay, so let us go in detail a bit. Okay, the same thing. Okay, so passive immunization. When can we give passive immunization? That is the three conditions. One is when the recipient is incapable of mounting an immune response in case of fetal or newborn babies. So in fetus and newborn babies, they are not capable enough to host or to mount a huge immune response. In that case, the baby is given or naturally the baby is provided with maternal antibodies. Or if in case the host is suffering from any immunodeficiency disease, so in that case from outside the antibodies needed to be given. So no time to allow the recipient to mount the immune response, as I have already told, like snake bite or any kind of insect bite. So if in that case we do not have much time in hand that the body will grow its own immune system or the body will grow its own antibodies. In that case ready-made antibodies has to be given immediately. Okay. So diseases for which antibody transferred are passively done. So these are some common diseases where passive immunization or ready-made antibodies are given, like botulism, tetanus, diphtheria, hepatitis A, B, rabies, snake and insect bite. So same thing. So let's move on. Okay, this is active immunization, as we have already discussed that active immunization can be coming from natural source, that is through infections or regular cold and cough, like that. Body is growing its own antibodies, and via vaccination. Okay, where vaccine from outside we are giving some microorganisms in dead form or weakened form, and our body is growing antibodies against it. So this is through active immunization. So let's come to the important attributes of an effective vaccine. So in order to qualify for an effective vaccine, it should be suitable for administration early in life. As we have seen that in just as the baby is born, we are given with a schedule of doses of different vaccines, like polio vaccine, like DPT vaccine. So there is a schedule of vaccines given, that is at early stage of life whether the vaccine is qualifying. So that is one attribute. Next, it should be affordable worldwide. Not that only a certain region is getting it, but it should be affordable worldwide. Next, it should be effective as far as possible after a single dose. Okay, always we have seen that in many cases, actually in many cases a single dose should be effective. Okay, not that every time we have to be given multiple doses, but most of the times it is that multiple doses needed to be given or booster doses are needed to be given, but single dose should be enough. So a single injection should contain vaccines for more than one diseases, that is combination vaccine. Okay, so if we are giving one single injection, and in that only we are giving the vaccine for many diseases. Should be effective when administered by mucosal route, that is through oral route, like oral polio vaccine. The requirement for cold transportation or storage shouldn't be rigid. Okay, the requirement shouldn't be rigid. That means it shouldn't be that the cold storage process should be a bit on the side that everyone should maintain it, or it should be maintained throughout the country or throughout the world. So requirement shouldn't be very much rigid, as we have seen in Covaxin or Covishield. It should induce both cellular immune response and humoral immune response. Okay, humoral, if as we have studied the previous units, humoral means antibody mediated immune response, and here cellular means T cell mediated immune response. Development of immunological memory is the ultimate requirement of all vaccine. So a memory cell or a memory should be generated. It can be of T cell and B cell. If the memory is generated, then if next time a same disease infects the same person, he will not suffer from the same disease again. So these are some attributes. Okay, so quickly we can see that recommended vaccines which occur in India or which operate in India, that is DPT, diphtheria, pertussis, tetanus, polio vaccine. They come in two forms. One is Sabin, another is Salk. Sabin is your oral polio vaccine, and Salk is the injectable polio vaccine. Then MMR, that is measles, mumps, rubella vaccine, chickenpox, Haemophilus influenza vaccines, and there are some other forms. Okay, so and there are some vaccines that are given during epidemic only or when it is required, not that it is given on everyday basis. So during epidemic or as required, doctors administer cholera, influenza, typhoid, rabies virus vaccine such as HPV, that is human papilloma virus, and as we have all gone through this stage, that is COVID-19 vaccine. So these vaccines are given when they are required or as situation arises, like in epidemic or pandemic situation. Okay. So vaccine scenario in India has improved a lot. Okay. So if we see in the 1970s, early 1970s. From that to 2021, the vaccine scenario has improved a lot. So it is that is the government is trying to cover a whole range or more range of people at the same time. So this is a schedule where we can see that vaccination is given at different times of the life stages, like at birth, at 6 weeks, at 10 weeks, 14 weeks. So different doses of vaccines are given at different stages of life. Mostly they are being given at 9 to 12 months, and then the booster doses started. So this is one thing. So moving on to the next slide. Types of vaccines. Yes, types of vaccines. So they can be categorized into several types. One is your live attenuated vaccine. We will be going through this in detail. First have a look at here. Inactivated killed vaccine, subunit vaccine, recombinant vaccine, conjugate vaccine, multivalent, DNA, recombinant vector, and edible vaccines. So here live attenuated vaccine, just we will go through this afterwards. Yes, live attenuated vaccine. That means in this case the microorganism that is being injected in the form of a vaccine is not dead. It is live, it is alive, but it is attenuated. That means its immunogenic or pathogenic capacity has been weakened. Okay. So in live attenuated vaccine, the microorganism like the bacteria or the viruses pathogenic capacity has been weakened by growing the bacteria or growing the virus in adverse conditions. So if we grow the bacteria or culture the bacteria in adverse media, what happens that their pathogenicity decreases. So in that way we can see that the vaccine, that is, it is suitable as a vaccine candidate, but it will lose its pathogenic capacity. So we can administer such vaccines like BCG vaccine, smallpox, oral polio vaccine, MMR, that is measles, mumps, rubella vaccine in the form of live attenuated vaccine, where the microorganism is not dead, it is alive, but its pathogenicity is not there, or it has lost its pathogenicity. So in such cases live attenuated vaccine have a good memory. That means if we give it in 1 dose or 2 dose, it will be effective. Next is killed inactivated vaccine. Killed inactivated vaccine, as you can see the name, the microorganism or the pathogen is killed. Okay, the pathogen is killed, the bacteria or the virus is killed by several means. Previously heat killed mechanism was used, but as if we provide heat, the protein coat of the bacteria or the viruses can denature. So nowadays we do not provide heat, but we use formalin to kill the bacteria or kill the virus. So in this way, if we kill the virus or the bacteria, it can serve as a vaccine, but it is in the form of killed inactivated vaccine. One good thing about this vaccine is there is no chance of gaining the virulence against. If we are administering any virus, that is killed virus, so it will not gain virulence again in the body. But the drawback is it needs multiple booster doses. We have to provide multiple booster doses to increase its memory capacity. Okay. So these vaccines contain killed pathogens, hence they do not replicate in the body. Okay. At least 3 doses of killed vaccines should be followed by a booster dose. So some examples of killed inactivated vaccines are typhoid, cholera, pertussis, pneumonia, rabies, hepatitis A, influenza. They are some examples of where we provide killed inactivated vaccines. Next is important group, that is toxoids. We have, most of us has gone through or we have been given tetanus toxoid. Okay, we have seen this in our daily life. So tetanus toxoid, they are a group of vaccines where we are not giving the microorganism, not the bacteria, not the virus, neither the virus. What we are giving is its toxin. Okay, so toxoids are modified toxins okay, the toxin of the that is being secreted by the bacteria, so that we are giving in the form of a modified form, which have retained their antigenic capacity but lost their toxicity. It will not if we administer a toxoid, it will not regain its property or it will not cause the disease, but it will just remain in the body and body will recognize it as a foreign material. So toxoids are modified toxins which have retained their antigenicity. So absorption of toxoid to a mineral carrier. So what we do that toxoids are generally given along with a mineral carrier, that is a protein carrier, which will enhance their antigenicity in the host. Okay. So booster doses is needed to improve the efficacy of this vaccine. Common examples, very common examples are tetanus and diphtheria toxoid. Okay. Subunit vaccine. So next, coming to the subunit vaccine, in this case we are not administering the whole microorganism, that is bacteria or virus is not given as a whole, but some purified macromolecules from the bacteria or virus is used as subunit vaccine. In this case we can give the bacterial coat or the protein that is present in the bacterial coat or the viral capsid layer which is causing the disease. So in subunit vaccine we are not giving the entire microorganism, but we are giving the portions, or you can say the macromolecules that are causing that particular disease. So examples of subunit vaccine are inactivated exotoxins and bacterial polysaccharides. So inactivated exotoxins, as we have seen toxoids, that is also an example of subunit vaccine. That is, the purified exotoxins are given in case of subunit vaccine, which are being treated with formaldehyde. Okay. So toxoids, as we have already studied, tetanus and diphtheria, and bacterial polysaccharide, that is the bacterial coat that is made up of lipopolysaccharide layer. This purified polysaccharide layer can be used as a vaccine against certain bacterial diseases like streptococcal pneumonia, Neisseria, that causes meningitis. Conjugate vaccine. So next is conjugate vaccine, where the vaccine or the microorganism is being conjugated to a protein carrier that will induce both the humoral and cell mediated response. Okay. So capsular polysaccharides of certain bacteria like Haemophilus influenza, which causes meningitis in children, is conjugated with a carrier protein like tetanus toxoid. So if we conjugate the proteins along with the vaccine, that will increases its immunogenic property. So this preparation induces class switching, it will generate B cell memory and cell mediated immune response. There are several other types of vaccine preparations also, like the recombinant protein or recombinant vaccine. So the vaccines which are produced through recombinant DNA technology are called recombinant vaccines, okay, where a particular gene, that is the antigenic protein that is coding for the antigenic protein, that gene is being taken and that is cloned in bacteria, yeast or in other type of cells like mammalian cells. Then from there a purified antigen is taken and it is administered to the patient, okay, or it is not only patient, it is administered as a vaccine. So in recombinant vaccines, recombinant DNA technology is used, where a vector, or we can see the antigen, that gene is taken which is cloned into a particular cell, which can be a bacterial cell, yeast or a mammalian cell. So examples are hepatitis B and pertussis. Hepatitis B is one of the first recombinant DNA vaccine. Okay. So surface antigen of hepatitis virus, so if you see the hepatitis virus, the surface antigen of the virus, that is HBsAg, that is hepatitis B surface antigen, it is the first recombinant vaccine cloned in yeast. Okay. So similar to recombinant vaccine, we can also see DNA vaccines. So DNA vaccines are just quite similar, where we are not taking the microorganism but the DNA of that microorganism or that antigenic gene of that microorganism, and we are inserting it into muscle cell. Okay, generally preferable is the muscular cell where it is the genome is being inserted. After it is expressed in the genome or it is expressed in the muscle cell, it is expressed in natural configuration, means like in the cells or in the muscle cells there are antigenic presenting cells which will engulf the antigen or which will engulf the DNA and which it will present that particular DNA. Okay, so prolonged expression of antigen inducing effective memory response, it is a good example, or DNA vaccine is a good candidate for a vaccine, and research is being going on in this particular sector where because it is proved successful in animal models like in influenza and rabies in animal models, but and test is going on for human clinical trials. Okay, it is on a trial mode actually. So recombinant vector vaccines, as we have already read this, we will not go in detail. Edible vaccines. So this is a completely new genre where we will see edible vaccines, that they are plant based. Okay, plant based. So plant means the plants which are transgenic. Okay, the plants where we can induce a pathogenic gene and we can make a vaccine out of it. Okay, so this is a completely new sector, and research is going on this edible vaccines which are produced from transgenic plants carrying pathogenic gene products. It is an emergent vaccine technology. If it is successful, then it can be cost effective and it is easy to administer, and also the storage facility or the storage criteria will be a bit on the easier side. So expressed protein products are heat stable, activating both humoral immunity and cell mediated immunity. So say there are certain plants mostly used as edible vaccines, or the plants in which edible vaccines can be made are tobacco, potato, banana, tomato and lettuce. So antigenic proteins expressed are for rabies protein, enterotoxicogenic E. coli, that is E. coli bacteria, hepatitis B surface antigen, rotavirus, measles and another Norwalk virus. So these are the pathogens for which edible vaccines are made in these plants. Okay. So this is a diagram we can see where viral vector DNA and mRNA vaccine, or you can see that as we have already discussed, like DNA vaccine, similar to DNA vaccine there is RNA vaccine. So this picture only denotes or depicts that mRNA is used as a vaccine and it is being encoded by a lipid sheet. Okay, mRNA is encased in a lipid nanoparticle coat, so it can enter the cell. In order to make its entry into the cell, we have to coat the mRNA in a lipid. Okay, then it is entering the cell. After it's entering the cell, the mRNA is going through transcription and the mRNA is going through translation, that is from mRNA protein is being made. After the protein is being made, it is expressed through antigen presenting cells and immune response is being seen, as we can also see with this replication defective viral vector DNA. Okay, so this is the diagram that is depicting all the vaccines I have just discussed, that is whole virus, inactivated killed virus, live attenuated virus, virus like particle, recombinant virus, DNA virus vaccine, recombinant vector vaccine, subunit vaccine, and there is also synthetic vaccine. So these are certain vaccines we have just discussed. Similarly, we can also see this chart where the types of vaccine, how are they working, their advantage, disadvantage, and certain examples are given. So we should know this. Actually, as we are studying vaccine, we should also know about the Covishield and Covaxin. Okay, so Covishield, that is being manufactured by Serum Institute of India, and Covaxin is also there. These are certain vaccines we are administered, which is administered for your COVID. Okay, and this Covishield is manufactured, which is used which is also used to manufacture, or sorry, the Covishield vaccine is manufactured by Serum Institute of India, who have they have also manufactured Ebola vaccine. Okay. So let's move on. So any vaccine, if you see, they go through several phases before human trial. Okay. So first is the phase 1 trial, next is phase 2 and phase 3 trial. Phase 1 trial is usually less than 100 people. That is, a vaccine is being tested on less than 100 people first to monitor the safety at multiple doses. If they pass the phase 1, then we can move on to the phase 2 trial, where the vaccine is being given to a larger mass of people and we see their effectiveness, okay, for a certain period of time. And then final is the phase 3 clinical trial. In phase 3 clinical trial, more than means thousands of people are given this vaccine at the same time to see whether it is fully effective or not, what are the advantages, or what are the, if there is any side effect or not. So in order to see this, the vaccine goes through these 3 phases or trials. And if it passes the phase 3, then we can give it in open market for selling. So there are certain challenges faced that for which vaccines cannot be prepared totally, like HIV. There is not a certain vaccine against HIV, okay, because there is a high rate of mutation in the genes. Okay. Next is rotavirus. Okay, rotavirus, there is no certain, there are certain vaccines, but they are not fully effective. Malaria. Okay. So in a year, thousands and lakhs of people died due to malarial infection, but there is no certain vaccines against these diseases, and the causes being the high rate of mutation, okay, immune evasion or complex life cycle stages. Like in malaria, the plasmodium, which is causing the malarial disease, it has a complex life cycle. So we cannot administer a proper vaccine against it. Immune suppression or intracellular forms. So these are certain criteria which we can see that poses a deep problem for causing or for creating vaccines against these diseases like HIV, rotavirus and malaria. So as we are giving vaccines to the children or infants, vaccines for adults are also there, like for meningococcal vaccine or your Tdap vaccine, zoster vaccine, pneumococcal and influenza vaccine, HPV or human papilloma virus vaccine. So what do we learn from here today? So this is the synopsis, or this is the summary we can say. So about the types of vaccines. So they are live attenuated vaccines, okay, for measles, mumps, rubella, certain things like that, and killed vaccines. Killed vaccines are given for polio, influenza. Toxoids are given for tetanus, diphtheria. Subunit vaccine, as we have just learned, that in subunit vaccine the whole organism is not given, but a particular protein is being given in the form of vaccine. Like here subunit vaccine example like pertussis, influenza. Next is virus like particle in HPV, human papilloma virus. Okay, like this the list goes on, like viral vector, nucleic acid vaccine. So this is the synopsis, or we can say the summary about the different types of vaccines and whether they are live attenuated or they are killed or they are toxoid or subunit, whatever the origin may be, but the main aim is same, to prevent a particular disease. Okay. And lastly, there are several generations of vaccines, like from the date of their origin or the date of their development. There are first generation vaccine, second generation and third generation vaccine. So first generation vaccines are created in the early 18th or 19th century, 1900s, that is, they are live attenuated vaccine and inactivated vaccine. They are first generation vaccine. Next is your second generation vaccine, that is subunit, toxoid and conjugate vaccine. And lastly, the third generation vaccine, in which research is being going on still, that is your DNA vaccine and recombinant vector vaccine. So these are the textbooks from which you can go through for this unit in your immunology. So I have given this. I am thankful to our principal sir, Dr. Monojit Ray, for giving me this opportunity to deliver a video lecture on this important topic of immunology. Also, I give my heartfelt gratitude to our zoology department of Barakpur Rashtraguru Surendranath College. Thank you.